In the previous lecture, we discussed photosynthesis. We discussed the overview of photosynthesis and the light reactions. And in this lecture, which is a continuation of the last, I'll be discussing the Calvin cycle. So in the last lecture, we learned about the light reactions, and this is where ATP is produced by photophosphorylation, and NADPH is produced as well. These are two energy-carrying molecules. And what we're going to focus on now is the Calvin cycle, which will use these two products of the light reactions, that is the ATP and the NADPH, and it's going to use them to power the fixation of carbon dioxide into an organic compound, specifically sugar. This Calvin cycle also takes place in the chloroplast, just like the light reactions, but the suborganellar location is different. Where the light reactions occur on the thylakoid membranes, the Calvin cycle is going to occur in the stroma. So the Calvin cycle doesn't require light at all, but it can't happen unless the light reactions occur because it is so dependent upon the products. So it requires ATP and NADPH from the light reactions, and carbon dioxide comes from just the air, the atmosphere, and it enters into the leaf through little pores, and this carbon dioxide is then fixed. So carbon dioxide is fixed into sugar, and we'll talk about the steps. One of the first steps is that it is going to produce a molecule called glyceraldehyde 3-phosphate, which is often abbreviated G3P. And this 3-carbon compound, two of them will be combined to make one glucose molecule. Remember, glucose is a 6-carbon compound. Now, producing a molecule like this, a biosynthesis like this, is going to take a lot of energy. So this is where the ATP comes in. ATP from the light reaction is going to be used during the cycle, and what will be produced will be then the ADP plus an inorganic phosphate. So that will have to go through another round of photophosphorylation to continue to drive the Calvin cycle. The other molecule is NADPH, which, if you recall, is a very similar molecule to NADP that is used in cellular respiration, and it is going to be used and form NADP+. So again, in the end, we're going to end up with a molecule of glucose. So we're going to go over the three major steps. We're actually not going to go into this in a great deal of detail. We're going to go over just highlights of the Calvin cycle. I'll first discuss the carbon dioxide fixation, then the reduction reactions in order to produce the organic molecules, and also then the regeneration of a molecule called ROBP, which is the carbon dioxide acceptor. So in carbon dioxide fixation, carbon dioxide is attached to a 5-carbon compound called ribulose bisphosphate. The reason why it's called ribulose bisphosphate is that ribulose is the name of the 5-carbon sugar, and it's represented here as just 5 circles connected together. And then this ribulose has attached to it 2 phosphates. Bis is another way to say 2. So all as this name means is that this is a sugar that contains these 2 phosphates on it. It's often abbreviated as RUBP. Now the carbon dioxide is going to attach one at a time. And when the carbon dioxide attaches to the ribulose bisphosphate, it's going to very briefly form a 6-carbon compound. And this 6-carbon compound with the 2 phosphates on it will very quickly split into 2, what is called 3-phosphoglycerate. So 3-phosphoglycerate is, you might recognize this name, because 3-phosphoglycerate is one of the many compounds in the process of glycolysis. But this is also something that is produced in the Calvin cycle. So this particular reaction is one that is catalyzed by one of the most inefficient enzymes on the planet. The name of the enzyme that catalyzes this reaction is called RUBP carboxylase. It is often referred to as Rubisco. It is so slow that the cell needs a lot of these enzymes. So it is an extremely abundant enzyme in plant cells and in photosynthetic organisms, and therefore is one of the most abundant enzymes on the planet. So carbon dioxide is fixed 1 CO2 at a time onto a 5 carbon compound to make a short-lived 6 carbon intermediate, which is quickly split into 3 phosphoglycerate. Just a note here, you do need to know the names of these intermediates in the process of the Calvin cycle and of carbon fixation. You also need to know the name of the enzyme that catalyzes this extremely important reaction for photosynthetic organisms, and therefore is important to you because you are very dependent on these organisms in terms of the food chain. So after the formation of 3-phosphoglycerate, there is going to be a reduction step. First, the 3-phosphoglycerate is going to receive a phosphate from ATP. It is going to be phosphorylated. And this phosphorylation product, which is called 1,3-bisphosphoglycerate, you do not need to know the name of this intermediate product, but this 1,3-bisphosphoglycerate is then going to be reduced. What will donate the electrons for its reduction is NADPH. And NADPH, in turn, will be oxidized back to NADP plus and can be reused again in the light reactions. So after this process of 3-phosphoglycerate being phosphorylated and then reduced, it is going to form a product called glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is often abbreviated G3P, and this name of this molecule shouldn't sound completely foreign because this is one of the products of glycolysis. So this same molecule that is now produced not by glycolysis, but the same molecule that's produced in the Calvin cycle, can then be used to produce the sugar. So G3P is then the base molecule by which, for example, 2 of them can be put together to make glucose, and actually all the organic compounds that a plant contains, every single one, its fats, its carbohydrates, its DNA, and its sugar, all come originally from this base molecule that's produced by the Calvin cycle and photosynthesis. The entire plant that you see, or all the organic molecules that you see in a plant, they are all produced from carbon dioxide. Plants are enormous CO2 sinks. What it gets from its roots is just water and dissolved minerals in that water. Okay, so let's do a little tallying here before we move to the next step. It actually takes 3 turns of this cycle to produce 1 net G3P. It takes 6 turns of the cycle. That means 6 times where carbon dioxide is produced to make 1 6-carbon glucose. But even with the number of turns of the cycle, on any given cycle, the original RUBP is split into 2 3-carbon sugars. One of them is spit out, but the other one, it gets to continue through the cycle for the purpose of regenerating that RUBP. So the regeneration of RUBP is a relatively complex series of reactions of which we're only going to go over the beginning and the end. It rearranges this glyceraldehyde, one of the molecules of glyceraldehyde 3-phosphate, and it gets rearranged back to ribulose bisphosphate. This process is going to then act as that carbon dioxide acceptor in the next round. Now, in order for this to happen, it requires a tremendous amount of ATP for energy, and that ATP also came from the light reactions. Okay, so let's look at this all together. Okay, so let's review this. The first step of the Calvin cycle is carbon dioxide fixation. This involves taking a 5-carbon compound called ribulose bisphosphate, and to it is added carbon dioxide to form a short-lived 6-carbon compound. This reaction is catalyzed by the enzyme Rubisco. This 6-carbon compound quickly breaks down to form 2 molecules of 3-phosphoglycerate. It takes 3 turns of this cycle to net 1 3-carbon compound. It takes 6 turns of this cycle to net 1 6-carbon compound. Now this molecule that is the result of the fixation, 3-phosphoglycerate, then goes through a reduction. This is the second phase. And in the reduction, the 3-phosphoglycerate is first phosphorylated by the use of ATP, and then the product of that is then reduced to form glyceraldehyde 3-phosphate. Both the ATP and the NADPH, which is the source of the reduction power, are products from the light reactions. Now, again, it takes 6 turns of this cycle to produce a glucose, which is a 6-carbon organic molecule. To produce 1 G3P, it takes 3 turns of this cycle. Some of the glyceraldehyde 3-phosphate, though, is used to regenerate the ribulose bisphosphate. So the last part of this Calvin cycle in review is the regeneration of RUBP. We didn't go over any of the details of these reactions, but G3P is going to be converted to ribulose bisphosphate, and this does require ATP. The ATP, again, one of the products of the light reaction. Once RUBP is regenerated, remember it's the carbon dioxide acceptor, so now it can then fix a molecule of carbon dioxide catalyzed by Rubisco and start this process again. So G3P has many fates. G3P is moved from the chloroplast to the cytosol, first of all, and G3P can be used to make all the macromolecules of the cell. It can be used to make carbohydrates. It can be used to make, and the carbohydrates, for example, are glucose, sucrose. It can make starch. It can make cellulose. Remember, cellulose is a structural polysaccharide found in plant cell walls. This molecule can also be used as the base molecule eventually to produce lipid. It is what is going to produce the nucleic acid and the protein. In other words, everything in a plant that is organic originally came from the carbon dioxide from the atmosphere. A tremendous carbon dioxide sink. But G3P, if you recall, is also one of the molecules that can be is a breakdown product of glucose in the process of glycolysis. So it can actually steal some of this and not use it for producing additional organic molecules at all. It can just take the G3P and just stick it right into cellular respiration and have it be converted to pyruvate and then regenerate ATP for the cell in the mitochondria. All right, let's do our final review of photosynthesis. Photosynthesis involves 2 major steps. The first is the light reaction, which requires light, and the second is the Calvin cycle, which is light independent. The light reactions happen mostly on the thylakoid membranes of the chloroplast. The thylakoid membranes contain the chlorophylls that are organized into photosystems, and you learned they were called photosystem 1 and photosystem 2. Now these chlorophyll molecules absorb and send that energy to the reaction centers, so the light is absorbed, and then it is used to excite electrons. The electrons are used to power the formation of ATP through the process of photophosphorylation, and eventually are transferred to NADP+ to make NADPH. Both ATP and NADPH are required for the Calvin cycle. The electrons, though, that are transferred to make this molecule of NADPH need to be replaced, and they're replaced in photosystem 2, where a molecule of water is going to be oxidized. That is, it will donate its electrons, and it will form as a product of that molecular oxygen. Now, molecular oxygen is not required for the process of photosynthesis, but it is used by another organelle, the mitochondria, which is found in plants, to power its cellular respiration, and that same molecular oxygen is also used to power your mitochondria to produce your ATP. So the Calvin cycle involves the production of the sugar, and it's it forms in three phases. The first is the carbon dioxide fixation, where carbon dioxide is fixed on ribulose bisphosphate to form a molecule of 3-phosphoglycerate. This is catalyzed by rubisco. So that is the carbon dioxide fixation. Then this molecule is going to go through a reduction. Actually, first it goes through a phosphorylation, then a reduction to form glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is what is used to form sugar. Two of them can form a glucose and all the other organic compounds in the plant. Some of the glyceraldehyde 3-phosphate, though, is going to be regenerated, and it is used for the regeneration of RUBP, and this process takes some additional ATP produced by the light reactions. The regenerated RUBP is used then for the carbon dioxide fixation on another round, and that is photosynthesis.